You should recall from your previous studies, the proteins are important as they make up around 18% of living cells. They are large molecules built of subunits or monomers called amino acids, which link together to form a type of polymer called a polypeptide. There are 20 naturally occurring amino acids which are either produced or consumed by humans. Proteins are very large molecules which can contain thousands of amino acids. These large molecules fold and organise into different shapes. What we are looking at today is that protein structure is described at 4 different levels of organisation: primary, secondary, tertiary and quaternary. This concept links directly to the 2013 biology study design. Unit 3, Outcome 1 states that the key knowledge is the structure of protein in terms of primary, secondary, tertiary and quaternary levels of organisation. This is Pat. He works for CSL, which is a biopharmaceutical company that identifies, develops and commercialises biotherapeutic products that prevent or treat serious medical conditions.
Hi, my name is Patrick James. I am a senior scientist at CSL Limited. Today I'm going to be talking about some of the work I do specifically on biotherapeutics, which are recombinant proteins which we produce for rare diseases. One of the roles that I have in my group is what's called mass spectrometry. Mass spectrometry is an instrumentation system that we have to measure the mass of proteins or peptides.
Before Pat explains his research, we need to understand the different levels of protein organisation, starting with the primary structure. Amino acids join together by forming peptide bonds, resulting in a polypeptide. The primary structure of a protein is a specific linear sequence of amino acids. Each protein has a unique primary structure and therefore has a special function. The order of amino acids in a protein is decided by the genetic material in the nucleus of the cell.
Primary structure is the linear sequence of amino acids which conforms a peptide. By using mass spectrometry, I can sequence these by selectively cleaving at the peptide bond and then comparing this back to databases of proteins which we produce. This is important for us to ensure that we are producing the correct protein which we then give to patients for their diseases.
Polypeptide chains can fold in 3 different ways. Hydrogen bonds form between nearby sections of the folded chain and help stabilise the 3-dimensional shape. The first example of a secondary structure is a beta-pleated sheet, shown here. This type of structure is found in silk molecules. The second example of a secondary structure is an alpha helix, which is a spiral molecule. This is the major type of structure found in wool. If any part of the secondary structure is not an alpha helix or beta sheet, it is called random coiling. Myoglobin, found in mussels, fits this category, as it has random sharp turns in its coil.
At the primary level, we have, as I mentioned, we have a linear sequence. However, for proteins to have function, they have to fold up. The first step of this is secondary structure. For me, specifically to the work I do, and specifically on antibodies, disulfide bonds are critical for secondary structure. The work I do at mass spectrometry is to map disulfide bonds to ensure that those that we produce recombinantly match exactly to those that are naturally found in humans. And this is important, thus that the protein works, but also that it doesn't affect adversely the patient.
The shape of the tertiary structure determines the protein's function. In enzymes, if the tertiary structure is altered, it will no longer perform its job, which can cause many problems. A tertiary structure indicates the overall irregular folding of a protein held together by hydrogen or ionic bonds, forming a complex 3D shape. These bonds are between side chains of amino acids and form complicated internal structures.
Moving on from secondary structure, we have the tertiary structure. Now this is the 3-dimensional fold of the protein. And this, like for antibodies, is critical for their function. For an antibody to bind to its target, it needs to have the right fold. To do this, I use a technique called hydrogen deuterium exchange, another form of mass spectrometry. This can be used to determine which parts of the protein are exposed to the surface and which parts are buried. And this is critical to the protein structure.
It is important to note that a quaternary structure cannot be described as 2 or more tertiary structures. The correct definition of a quaternary structure is that it contains an interaction between 2 or more polypeptide chains. There are many forms of this, including globular, as in hemoglobin, or fibrous, as in collagen.
Antibodies don't come as a single polypeptide chain. There are actually 4 polypeptide chains held together by disulfides, and this can be considered its quaternary structure. An antibody is made up of 2 heavy chains and 2 light chains, and the very terminus, the end terminus of the heavy and light chain pair on each side determines the actual binding capacity of this antibody. This is critical for the antibody's function. This work, again using mass spectrometry, but also using a technique called X-ray crystallography, we use to determine what the actual quaternary structure of the protein is, and we compare this we make in-house to those that are deposited from other antibodies which have been analysed from humans.
Prions are a type of pathogen that causes neurodegenerative diseases in mammals. Unlike regular proteins, prions can exist in two different conformations at the level of secondary structure. There is an alpha helical portion of the normal protein that is refolded into the beta sheet formation in the abnormal protein. The primary structure of the prion remains the same, but its secondary structure is different. As a result, its tertiary structure will be different as well.